Hello dear students, I welcome you all to this first online lecture, an initiative taken by our Honorable Director of SCIMS and on orders received from our principal. This will be the first lecture followed by a series of lectures in order to compensate for the classes we have lost during the lockdown. Today, the class is for MLT second year. This is the first lecture and it's about Introduction to Microbiology and Historical Perspective. So let's begin. When you talk about microbiology, the first thing that you need to look about whenever you study anything is terminology. So when you look at this microbiology, you will see that it has two different terms. One is micro, which stands for small, or it stands for 10 to the minus 6 when we talk about it in the numerical form. And biology means study of life. So basically, microbiology is the study of small microorganisms that are not visible to the human naked eye, and it then includes two things. One, to study different kinds of microorganisms, and second, how to culture them, the different techniques involved in culturing of these microorganisms. So this domain of the study is called microbiology. So we can say that it is a study of these biological organisms which are too small to be seen with the naked eye, but with the help of magnifying glasses, as were used by the initial microbiologists, or after the invention of microscope, it helped us to reach and see how important these microorganisms are. There is no historical account of who was the first one to start studying the microorganisms. But before I begin this lecture, let me make it very clear to you that microbiology is not a new field. The people of Roman empires have been using unknowingly the microorganisms, for example, making for yogurt, cheese, and beer by the process of fermentation. And then a process came. With time, we started to understand them and correlate them with the diseases. That was called germ theory. And after then, to complete the circle, we also understand the importance of these microbes in our day-to-day medical life. The people who are suffering from diabetes, it's the microorganisms, and with the help of the modern techniques called biotechnology or genetic engineering, scientists have been able to produce insulin that has saved so many lives. We also get vaccines from them. So microbiology, or the microorganisms, have both the phases, the beneficial phase and the pathogenic phase. So if we try to divide these organisms, we can divide them mainly into four groups. The first one is the viruses. We don't consider them as living entities. Outside the host, they are called viroids, in the sense that they have a nucleic acid, a genetic material, surrounded by a protein coat. So generally they are like packets of nucleic acids surrounded by a protein coat. And if they get a host, they start replicating. So viruses stand at the borderline of living and non-living. Coming to the next group, we have bacteria. We have the good bacteria, we have the bad bacteria. We have the normal microflora of our gut, we have the skin microflora that are beneficial to us. But at the same time, if these microflora enter our body on the other side, then they become the pathogens, and our body immediately starts responding against them. So based on the gram staining, we differentiate the bacteria into gram positive, gram negative, and there are some bacteria, for example Mycobacterium tuberculosis, they don't fall in the category. So usually they are being stained by Zeal nasal staining. I'm talking about MTB. So this bacteria is like a fungal. It's a fungal, fungus-like bacteria. It means it's showing it's showing both the characteristics of the prokaryotes as well as eukaryotes. So if I borderline it here, the fungi and the parasites, they belong to eukaryotes. And what are eukaryotes? Eukaryotes, eu means true and karyon means nucleus. So they have a true nucleus. It means they have a nucleus in the cell, and then they have their membrane. They have many different cell organs inside. Compared to the bacteria, the structure is very simple. They have the nucleic acid, okay? So they have the genetic material, but it's not contained in the nucleus. They don't have any cell organelles. Yes, they have respiratory chains in the form of mesosomes. So these are the four major groups which we study in microbiology. So when we try to classify them, we have a plan of hierarchy which we use for every organisms, and that hierarchy stands from domain, then kingdom, and then it follows the process of class. Imagine you are sitting in a class and you are sitting in a order on the benches and you belong to different families, okay? And then definitely we have the species, I mean the genus first and the species. So we have domain, kingdom, and then we have different phylums. So then we enter the class, the order, the family, the genus, and the species. So what actually this means is it's like a passport of these organisms. If you look at the passports of them, you understand where they belong to which family they belong. But when you talk about groups, so we have these four groups: viruses, bacteria, fungi, and parasites. Now, when we try to classify these viruses, we can classify them on different bases. In case of viruses, we can differentiate them based on the nucleic acids. They can either have RNA, that is ribonucleic acid, as a genetic material, or DNA. Best example is, for RNA viruses, is your coronavirus. Coronavirus has wreaked havoc right now around the world, and it's pandemic. It means it is all the countries are suffering. Then we have also DNA viruses, like herpes simplex virus. It causes cold sores on your lips. We also have HIV. It's an RNA containing virus. Then we can further differentiate them on the basis of the second criteria is the lipid envelope. Just as cell membranes have a phospholipid bilayer, the lipid is same, the envelope is the same, and it has been many scientists have pointed out that this lipid envelope comes in, they have adapted because once they entered into the animal cell, while going out of the cell, then they take the envelope along with that. So it's very similar to your phospholipid bilayer membrane, which is present in our cells. It means human cells. So taking advantage of this coronavirus can have this envelope and it interacts with our nasal epithelial cells, and then it can cause swelling in the inflammation in our lungs and causes multiple organ failure. So next we are talking about bacteria now. Bacteria are prokaryotes. Based on the gram staining, they can be differentiated either gram negative or gram positive. They belong to the category of prokaryotes. Pro means primitive, karyon means nucleus. So they don't have nucleus usually. No any other organelles as well. One example of gram-negative I can give is E. coli. E. coli is one of the well-studied bacteria in microbiology, and it has helped in understanding the mechanisms of genetic engineering. And one of the examples of gram-positive is B. subtilis, Bacillus subtilis, and they are spore-forming bacteria. Coming to the third category, we have fungus, or fungi. They can be differentiated based on if they have a single cell or if they are multi-celled. The best example of single cell yeast, that is S. cerevisiae, a baker's yeast, and we have S. pombi, Pombi. So this is the species, and this is the genus, and this is the species. Okay, as I told in the hierarchy. Coming to multi-celled cells, we call them as molds. You will see when you have a bread left out in the fridge unattended, and you will see that there are some different variety of colors coming up like this on your bread. So these are molds. They are multicellular. Of course, they are eukaryotic. Coming to the fourth category, that's the parasites. The single-celled are called protozoa. The best example of protozoa is the Plasmodium falciparum, that is causing malaria. Okay. Now, coming to the multicelled worms, we have flatworms. They're all helminths, and we have roundworms, which you will find particularly in the childhood, and we have been given medicine against it, where I have received myself. We have flatworms. And we have also this Tinea which is present in the pigs. And if consumed without cooking properly, the pork, then it goes to your brain. It causes cysticercosis that can lead to the or they can trigger epileptic seizures. Then the third category of parasites is bugs. To be clear enough, I have divided them into very simple forms like ticks and mites. Mites destroy the furniture you have in your house. Then we have mosquitoes and flies. Mosquitoes can cause malaria because they are the carrier of Plasmodium falciparum. And then we have flies that can contaminate our food. Then we also have lice, which is nowadays very rare to find in humans, and fleas. Okay. Now moving to the next slide. Louis Pasteur. We are already aware with this technique called pasteurization. We have heard a lot about it in the dairy industry. So in order to protect the dairy from the bacteria or any other microorganism, Louis Pasteur gave an idea of pasteurization because it was him who gave this idea. And his early work has started in the 18th century, in the mid of it. So he said that microorganisms can they can do fermentation, and in case of mussels we form lactic acid, but in case of pyruvate, if oxygen is not available, it can either change into lactic acid or it can change into alcohol. This process is called fermentation. It's an anaerobic process, but we also use fermentation on the industrial scale. The term is also used on the industrial scale to manufacture something from the microbes in a large quantity. And for that we need fermenters. Okay. Now, it was 1861 when Louis Pasteur rigorously disapproved a concept called as spontaneous generation, which I will talk about in the next slide. In 1881, he developed the first vaccine against anthrax spores, and you might have heard about bioterrorism. These spores were packeted in an envelope and sent to the president of USA. So the bioterrorism concept came from this. In the late 1885, he also developed rabies, that is an RNA containing virus. And what are these vaccines? You might have heard today we have a lot of talk about coronavirus vaccine. Why don't we develop a vaccine against coronavirus? And why is it not so easy to develop a vaccine? So the vaccine comes from the word vacca, which means cow, and in honor to respect of Edward Jenner, who first developed the vaccine against smallpox by using cowpox virus. So we call it a vaccine. A vaccine is like if you have a bacteria or a virus, you can either kill it, okay, you can kill a virus or a bacteria, or you can attenuate it. So we have killed bacteria, or we have attenuated. Attenuated means you weaken its pathogenicity, but you can use it as an antigen. Once you take it as an antigen, you can add to it an adjuvant in order to make its molecular weight and prepare a vaccine. It means you are alerting your immune system before if this organism is going to cause disease to you. So in order, we are teaching them, or it's like a training, a pre-training. If there is any attack on any camp or something, so they pre-train them. So they are called drills, mock drills. What is a mock drill? Mock drill is to activate your forces, to train them that if the actual attack happens, they can immediately make a response. So that is about the vaccines. Coming to the next. So there's no doubt that I forgot to mention that that's the reason why, because he has been working for so much time, he is rightly called as father of microbiology. Coming to the historical perspective, it was very unsure that who was the first to observe the microorganisms until the discovery of the microscope itself. And during this 15th and 16th, early 16th, it was Jensen who developed the first compound microscope. And what is compound microscope? You know, microscope is something like telescope. Telescope is showing us far away things, and microscope can show us very small things, which are of size of minus 6 microns. So compound means it is a system of two lenses, one lens and another lens. Or you can simply see microorganisms using a magnifying glass. That is what earlier microbiologists used to do. They have magnifying glasses with them. So compound means is a combination of two lenses. For example, we have an eyepiece, that's called one, and then we have objective lens. So when we have a system of two lenses, this is called compound microscope. It was 1665, that's very early, an English scientist by the name of Robert Hooke. He discovered cells, and that is it is a major breakthrough of that time. And he says that actually what he was saying is, it was not the cells. What he was seeing is the dried cell walls of the plants, a plant cock. And he's also reputed to have observed some strands of fungi based on his, maybe some help from Jensen in providing him this microscope. So he also saw some strains of fungi. But it was in 1676, after Robert Hooke. It was A.W. Leeuwenhoek. He was a Dutch merchant, but he was also he was also born with a scientific temperament. So he started carefully observing what are present in the droplets he collected from a pond. So in that pond he saw something different things, and they were trying to move around. So he named them as animalcules. They are animal-like because animals move. And then, because of this, he is regarded as the first scientist that has provided accurate descriptions of bacteria, fungi, and the protozoa, as I talked about, Plasmodium falciparum, which causes malaria. And he thus revealed, he was the first person to reveal the microscope world to the scientists. So probably he might be the first to do it. Next. Now, coming to the theory of spontaneous generation. What is spontaneous generation? Spontaneous generation simply means that you can produce something from non-living to living. From non-living matter, living things come into being. So what happened was, after Leeuwenhoek passed away, the interest in microbiology was lost, because most probably the microscopes were not that much readily available. They were very rare. Only few of the scientists had a real compound microscope. And it was Aristotle who provided this doctrine, because being a great philosopher, he gave a doctrine of spontaneous generation. That is, from non-living can rise living things. So what he did is, he made a beef broth, means he made a soup out of the beef. That's called broth. Broth means liquid. And he said, in this liquid, if you allow it to stand of its own, open, then you'll find small maggots developing on the broth. So this is what he was talking about. But later on, other scientists debated over it. We will see it in the following slides. So in the following years, it was debated around that time, and it was a debate never ending. So who are the people who are involved? Mainly, I will talk about them who were majorly involved in these debates. So in the end 16th century, it was F. Ready who first challenged the theory of spontaneous generation. And he showed that if raw meat jar is covered, the formation of maggots was prevented, that I have showed in the last slide. In 1748, it was Orr Needham. He was a preacher, and he tried to support his doctrine of spontaneous formation. He was in support, in favor of the debate. So F. Ready was against. R. Needham was in favor, and in 1776, L. Splancy challenged this spontaneous generation, and as he pertained to microbes, showing that if a container is sealed, and they were boiled, it means you are sterilizing your medium. There was no production of maggots. The microorganisms were not found. And then in the 1858, Rudolf Virchow gave a very challenging and a different kind of view. He was a German scientist. Most of the research was initiated in Germany. Most of the discoveries come from Germany, and then the US exploits it further for their own benefits. So he challenged it, and he gave a concept of biogenesis. What is biogenesis? Biogenesis simply means all cells arise from pre-existing cells. It is a statement which you might have studied in class 12th. It was Rudolf Foucault himself. And probably all cells arise from placing cells by a process of cell division. In case of prokaryotes, the cell division happens by binary fission. And in case of eukaryotes, it happens by mitosis, because they follow a 24-hour cell cycle. And of course, the father of microbiology, 1861, he rigorously disapproved spontaneous generation, but with no experimental work being done. So then he decided to prove it experimentally. So he was challenging it, and now he has to prove it. The people were demanding proof from him. So he had disapproved, and in order to validate his point of view, he has to do something. So he devised a series of experiments which are very famously known as Swan-Neck Flask experiments. So he said that the air surrounding us contains the microorganisms, and these microorganisms can come in contact with the broth and it can contaminate it. Okay, so he devised this neck-like flask. He said if the air goes in, the bacteria will be trapped inside this neck and the broth will remain clear. It means it will not develop turbidity. And turbidity is an indication of either overgrowth or contamination when you are doing microbiology in your practicals. So he left the flask of broth open to air, and as can be expected, if the neck is not there, so contamination did happen. The broth does change its color and becomes turbid. And turbidity is a sign of growth or contamination. So because of the curvy neck, as I told you, no broth was contaminated. It was not contaminated. So the flask did not become contaminated as he predicted, and Pasteur's experiments put rest to their debates, and there was no one left to support this spontaneous generation, or the notion of spontaneous generation itself disappeared. Coming to the next slide. Now, as we talked about the good microbes and the bad microbes, so people started thinking that a degraded term using germs or bugs, people started thinking that they are the reason of many diseases. So it was Louis Pasteur, again the father of microbiology. He was working in the late 18th century, he performed numerous experiments, which we have already seen, the neck experiment. So now he was working, an industry invited him on a job. They hired him, and they had the problem that their wine and dairy products were becoming sour. They were becoming sour, and when they are sour, they become inedible. And it is a huge setback for the industry. So when they set him on finding out what is the problem, so he blamed bacteria for this problem. And Pasteur called attention of importance of microbes now in everyday life. It means what the Roman people were doing, preparing curds, preparing different kinds of dairy products, preparing beer, as they are very fond of taking beer, preparing alcohol. So now he made the scientists understand that it's all the microorganisms actually that are doing this work for us. And on the other hand, he stirred the scientists thinking that Many of the human diseases can be linked to these microorganisms, because if it can make a wine sick, definitely it can create or make a human sick. And his work encouraged that belief that microorganisms that are present in the air could cause diseases. And some of the things you can understand here is air. Air, you can see that in case of tuberculosis, if somebody with tuberculosis sniffs or he sneezes or he coughs, the droplets are produced. These droplets are taken by the normal human beings and they get infected. It can be same will be the case with the coronavirus. Because of the coughing, small aerosols can be generated and a nearby person can take it in and get infected. So he was right in the sense, saying that yes, they can cause diseases also. But he said they are present in the air. But we know all the microorganisms are not present in the air. They can be in the soil, they can be in water. But this was a very good scientific prediction. So he postulated what we know today as germ theory of diseases, that germs can cause diseases, that is, microbes can cause diseases, and microorganisms are causes of infectious diseases. So we can get different diseases, but what is the difference between disease and infectious disease? Infectious means if it spreads to one person to another person. So TB is an infectious disease. HIV is an infectious disease. It can spread from one person to another. Same coronavirus, COVID-19, that the strain that developed in 2019 December, although it had also emerged before, but no work was being done. It emerged as SARS in China in 2002, and after 10 years it emerged as MRSA, Middle East coronavirus, Middle East respiratory MRSA. It was 2012, and imagine within the next 8 years it has erupted again as COVID-19. So it is mutating very fast. That's why we are not able to develop a vaccine against it, and it has many strains. And you will be surprised to know that COVID-19 is responsible for common cold, which we normally have. So imagine now a common cold can be associated with COVID now, because the symptoms are same. So it's like a confusing for scientists. That's why we are suffering like this. So there are so many deaths. Next slide. Now, again, after Louis Pasteur, I think the one person who should be credited with, giving a new refreshment to microbiology was Robert Koch. Robert Koch is credited for discovering Mycobacterium tuberculosis, responsible for tuberculosis. See, so Mycobacterium is the genus and tuberculosis is the species. We also have Mycobacterium leperi that causes leprosy. Okay, so this Rudolf Virchow's idea of cells arising from pre-existing cells, same is the case with bacteria, fungi, and the bacteria, fungi, and the parasites. Okay. However, it was German scientist Robert Koch. He was from Germany again. See, German people are very genius people. He provided the proof by using anthrax bacteria, which is a spore forming bacteria, and separated it from the other microbes that were present. The experiment I will show in the next slide. So what he tried to do is he tried to obtain the pure culture from the rest of the bacteria. Imagine that this is one kind of bacteria, this is another kind of bacteria. So they are in a mixed environment. So what he did is he tried to isolate the one that's causing the germ, that's causing the disease. Okay. So it means he has produced the pure cultures, and then he injected them into the mice, and he observed a very good results, which I will show in the next slide. And he gave some postulates which we call as Koch's postulates. Okay, so these are very important because they have now become as a framework for any of the diseases we are using in the microbiology. They provided a set of principles, so where we can say that which microorganisms is responsible for which kind of disease. Okay, next. So let us look at the Koch's postulates closely and look at this diagram. So this horse is suffering from, for example, say anthrax, and he isolated the causative agent, let it be anthrax, because this horse can contain different kinds of bacteria, and this V-shaped bacteria is your anthrax. Okay, so he was able to purify and prepare a pure culture. By preparing a pure culture, now he infects this pure culture and injects it into the mice, which is a healthy mice. Okay, the mice become sick because anthrax has a hold on him, and finally he dies. And the next thing what he did is he wished to see if this mice, from his biopsy, he could obtain the same bacteria again. So he said, okay, this is the bacteria which is killing it, and he tried to culture it again to see, to confirm, to verify that this is the same anthrax which he has injected into the mice and recovered back from the dead mice and cultured to verify. So the steps of Koch's postulates you can remember like this: 1, 2, 3, 4, 5, 6 steps. Okay, so microorganisms observed in sick animal, that's here. Cultivate them in the lab, that is here. Then inject into the healthy mice, that is step C. The animal develops the disease and dies or becomes sick. From that sick patient you can take the biopsy and you will observe those microbes in your culture, that is the plate culture. So you'll be learning different culture techniques using liquid broths as well as the plate culture techniques to culture different microorganisms, including viruses. Okay. So and then finally to re-isolate into the lab and see the same bacteria which we have observed here is coming out from the biopsy. So it's like a full circle now complete. So these were the Koch's postulates. Now, then after these things, when everything was over, that spontaneous generation was finally, the notion was put to an end, and a germ theory was put in place, and that led to the golden age of microbiology. So the development of this microbiology began in the late 80s for the first decades to 19th centuries, and scientists couldn't stop themselves to seize this opportunity further to see which kind of bacteria can cause which kind of a disease. And as such, this period is called as the Golden Age of Microbiology, and many infectious microorganisms were identified which used to cause particular kind of illness or a disease. So many of the etiological agents, the causative agents that were causing the one particular disease were found. Okay, and this led to halt in the epidemics, and then emerged the field of antibiotics. Now when we learn that this particular bacteria is causing this particular disease, now we have to study the mechanism of action of this. How does it infect the microorganisms, and do we have any defense against it? Yes, it was the first antibiotic that was discovered was penicillin. It was identified from a mold called Penicillium notatum, and it proved very beneficial in World War II. It saved the lives of many of the soldiers that were involved or got infected from, say, the bullet injury Okay. So what antibiotics is, anti means against, biotic means bacteria. So one bacteria can provide something to us that can kill the other bacteria. So we call them antibiotics. One bacterium or one microorganism killing the other. And it's perfectly all right, because there is a competition in the niche, because many bacteria live in the same niche, so they produce these secondary metabolites which acts as toxins and repels the other bacteria from their territory, so that they can have all the nutrients to themselves. The same is case with the infectious diseases as well. So thank you for patiently listening to this lecture, and if you have any comments, if you have any questions and comments, kindly send them to our WhatsApp group, which you are already you are already enlisted in. So see you again in the next lecture. Thank you very much.